Biological Sciences Fellows, which I've really enjoyed having here at VBI because we have a lot more wet lab people, so I can pop in and borrow reagents here and there. And with that...
He does actually have a sign that says 2 minutes. We have one simple question in my lab, or one simple goal in my lab. We just want to end cancer, hopefully before I retire. And I do believe that my generation is going to accomplish this. We have tremendous progress done in this area of cancer research in the last 40 years. We have tremendous work done in the genomic area to understand how the disease happened. And my lab is interested in understanding how environmental factors influence cancer incidence. So I think everything together can really have a big impact on cancer prevention. So we focus on environmental factors, and in particular, we are particularly interested in understanding how disruption of our normal physiology leads to cancer. And I'm going to push forward. Yes. And in particular, we are interested in understanding how circadian factors influence cancer incidence. And the reason because we are interested in circadian factors is because we know that when there is a circadian disruption, there is a chance of developing diseases like cardiovascular diseases like cancer. And this happens because your physiology is altered and is not working in the normal 24-hour cycle. For those that do not know very much about what circadian rhythms are, let me give you the 101 version, which is a model in which we have 3 main components. One is an input signal, and that could be light or a change in temperature. There is an endogenous oscillator in your brain that is the suprachiasmatic nuclei that captures those changes on the environment, and that leads to an output signal, and an output signal could be anything from waking you up in the morning, rising your cortisol levels, triggering melatonin release, and telling your body that you had to go to sleep. So this is a very simple mechanism in terms of concept. It's very complicated at the molecular level, of course, but the idea is that if you mess up somehow any of these components, you will end up with an output pathway that is not what you want and might end up developing a disease. So my lab is a lab that focuses mainly in cell and molecular biology. And what is particularly appealing about the circadian rhythmicity system is the fact that it's driven by synthesis and degradation of macromolecules, like very many of the typical biological processes. But why is that circadian rhythms have become so important lately? Well, one of the reasons is because, they are involved in a number of diseases and disorders. We know nowadays that they are arranged by shift work. We know that they are involved in alcohol consumption, in coronary heart attack, metabolic diseases, mainly in depression. And from the standpoint of our research, we know that there are important endogenous factors that contribute to cancer development and progression. We know nowadays that women that work night shift have a higher propensity to develop breast cancer and men prostate cancer. And this is because it's a hormone-related cancer mainly, and your hormones are totally messed up if you work at night, okay? This is really the person to blame on this is Edison, because he invented light. And when he invented light, he really screwed our lives. So now you don't go to sleep at 6 p.m. or 7 p.m. when it was dark. Now you stay working late at night, you work in your computer or you watch movies, and that causes what is called a social jet lag in your physiology. Okay, so you go to sleep later, you wake up at a different time of the day, not at the sunrise, and that keeps you out of synchrony with the environment. So all this causes diseases. So it wasn't that good to invent electricity after all. So why is all this, how is all this driven? Well, all this is driven by molecules that are called circadian factors, and these circadian factors are basically transcription factors, factors that control the expression of genes, turn genes on and turn genes off. They all have really cool names like period, double time, clock, and everything. And they are all arranged in a number of connections that actually drive the rhythmicity of very many genes involved in cell cycle progression and cell death processes. And we know nowadays that around 10% of all circadian control genes control cell divisions and death. Why is this important? Well, cancer has been totally defined as a disease in which you have uncontrolled cell progression. So if you have an environmental factor that tells the cell divide, because you get light every day at abnormal time, then the cell gets the signal divide, 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 and keeps dividing when it shouldn't be dividing. And that's what actually causes an hyperplastic process, for example, and then might develop cancer. So another way to see this is that the circadian factors control cell death processes. So one of the backup mechanisms in cells when something goes really, really wrong is that the cells are very altruist and say, well, hold on, there is something going wrong in me. I better die than keep dividing and propagate the mutations, right? Well, cancer is a very smart disease and control the cell death process. So tell the cell, nah, I was joking, just keep dividing, okay? So that's how things work. So let me, Jason, you think you can put that movie? So this is a very quick way to actually graph what cancer is. So you have one cell that is abnormal because it accumulates a number of mutations and divides at expenses of other cells in an organ. And then what happens is that you end up with cells that accumulates, pops up, and forms a small tumor. And this tumor will keep growing, recruit blood vessels, and when it recruits blood vessels, it will bring more food, nutrients, oxygen, and that will keep this tumor growing to the level that you don't really need a microscope. You will see it with your naked eye. And when this happens, eventually one of these cells in the tumor will skip the tumor, will go through the blood vessel, migrate to some other places, and eventually leave the blood vessel, like, for example, here, causing a process called metastasis and the presence of a secondary tumor. So why is all this important? Because we ask ourselves whether there is any chance that cell proliferation and thereby cancer can be a consequence of an abnormal circadian regulation. If this guy here controls cell division and cell death, maybe the imbalance of this due to abnormal circadian regulation can end up being cancer, can produce cancer. So the lab focus in two areas. One is prevention and early detection, and the other one is treatment. And why is this? Because I think the best way to prevent cancer, the best way to eliminate cancer is by preventing cancer from happening or detected at such an early stage that all what you need to do is a minimum invasive procedure to stop the disease, okay, or control it in a chronical way. So one of the areas in my lab works in linking at the molecular level how the circadian factors and the intracellular network works, which ones are the features of this interaction, which ones are the outcome of these interactions, how this controls cell proliferation, induces and controls the immune response that we actually see diminish in cancer patients, replication and angiogenesis, all of which are important for cancers to oh my gosh, 0 minutes? Oh, okay, anyway. So the whole point of this is that we use a combined system of approaches that will end up in helping us to develop better tools for screening, okay? And the main idea is that we want to develop a screening tool in which we can predict in advance what kind of things can go wrong and what patient is at risk of developing a disease, so we can change the circadian rhythmicity or the environmental situation and prevent the disease from happening. And in order to meet with the time deadline, I'm just going to tell you that on the treatment area, we have a very interesting project ongoing with clinical physicians in which we work in treatment options in which we link circadian rhythms to very many outcomes in treatments to find out which ones are the better treated strategies. So I'm going to stop here for the purpose of time, but we do have, like Chris says, evolve or die, so we decide to evolve. And we have incorporated a number of omics projects in our more recent work to understand how this large-scale analysis can help us to detect the disease early on and prevent it from happening. And this is my group, and this is where we live. Thank you very much.
I think we have a common theme in this symposium, and that is we talk about large and complex networks on all sorts of different scales. And for the next few minutes, I want to take you down to a very small scale, about 1/25th of a millimeter or smaller, and that's the world of cells, which also contain highly complex networks. All life on Earth is made out of these cells. A human being needs about 13 trillion of those. They can also work on their own. They can make all these other structures. And what's amazing is that a cell is nothing else than just a bag of molecules that interact with each other, and they interact with each other in highly complex ways. This is the protein interaction network of a quite simple cell. You see the cell in the middle here. This is about 5,000 proteins, often interacting with many others. And this is static, but of course the whole thing is very dynamic. And whenever we try to study signaling pathways in the cell, we actually find that it's frustratingly complicated. It's highly complex. So in general, in engineering, there's this principle that complexity is the enemy of reliability. Yet cells actually have both aspects. On the one hand, they are very complex in their regulation, and on the other hand, they execute this really reliably. And we are interested in this: how is reliability and robustness achieved in cellular function? And to study this, we look at a process that is essential for all life, that is highly dynamic, that is precisely timed, and that indeed has a complex regulation. And on top of that, it's beautiful to look at, and that's cell division. And what you just saw here is what normally goes on for about an hour, which was a mammalian cell dividing. We study this not in these mammalian cells, but in a simpler cell type, yeast, which, however, serves as a very good model for many other cell types. And we do that because we are interested in the basic regulatory principles. And that is much easier to understand in a simple cell type than in a metazone cell type, which has lots of added-on regulation that adds to the complexity and makes it even more difficult to understand. The imaging, as you see on the right side, is not quite as nice as it is in the mammalian cells, but we can image with a resolution between seconds and hours all the processes that we are interested in. When we study a process, we use whatever technique seems useful to us in order to address the problem, and we usually initiate collaborations to do that. So in the past, we have, for example, used chemical biology tools. We have used quantitative proteomics and phosphoproteomics. We have used even more advanced imaging than we can do in my own group. But one technique that we come back to again and again is computational modeling, because it's really helpful to understand how cellular signaling is wired. So we typically combine our quantitative experiments with a computational model for the process we are interested in, and then derive predictions from the model and test those predictions again in experiments. And we like it if the model works. We actually also really like it if the model doesn't work, because that tells us that we have not understood something fundamental and there is actually something to be discovered. So within cell division, we mostly focus on how do cells get the right number of chromosomes, which obviously is very important for the survival because they have all the genetic information that the daughter cells need. So we look at a signaling pathway that can detect if there's any problem with the chromosome attachment to the mitotic spindle, which would prevent them from being properly segregated. And we look at the process itself, how the chromosomes split and how they become distributed. And that also very closely links to the work of another VBI biology fellow who is not here today, unfortunately, Daniela Cimini's group, because they are interested in what happens to the cells once the chromosomes have missegregated and the cell ends up with a wrong chromosome number. So over the past years, with respect to the signaling pathway, my group has identified new links in this pathway. And as I told you, we are interested in how can it function reliably. And we have found that this pathway can only function reliably if protein levels are pretty constant. They do not fluctuate too much, and we cannot modify them much, otherwise the system breaks down. In this signaling pathway, we have also found other threats to its reliable function. which were surprising, but which are probably coming from a trade-off, because the pathway needs to be reliable, but on the other hand, it needs to be dynamic, and you cannot do everything at once. With respect to getting the chromosomes into the two daughter cells, that's completely different. So in that case, this step in the cell division is completely robust to large changes in the protein levels, and it was really difficult initially to understand why that is. And we have now started to figure out some of the mechanisms that allow cells to cope with such high variability and nevertheless really reliably deliver the chromosomes. So I've told you that we are interested in this aspect of robustness. How is cellular function reliably achieved? The flip side of that is the fragility. So in which aspect are cells or cellular function sensitive? And that links to two other areas. One is synthetic biology, because that tries to engineer new function inside cells, which often can be done, but often turns out not to be very reliable and robust. So very often does not work in different cell types or does not work in different environments. And we hope that what we find can inform these synthetic biology approaches. On the other hand, finding out where cell division is sensitive to perturbations can be very informative for cancer treatment, because as Carla already told you, cancer is basically just a disease of uncontrolled cell proliferation. So I would really like to thank my group, all our collaborators, all the other VBI biology fellows here at the Institute, in particular Daniela Cimini's group, with whom we have joined lab meetings. And our interests also really are closely aligned with the scientific interests of John Tyson at the Department of Biological Sciences, and Jean Pecou and Neil Adams here at VBI, with whom we also have joint meetings. And I would like to thank all my colleagues from the third floor and all the VBI staff who really make it fantastic to work at this institute. Thank you, and thank you for your attention.
All right. First of all, I would like to thank the organizers of the BBI for giving us a chance to see what we're doing. And I'm going to go with even higher resolution. Silk talked about cell size. I'm going to talk about protein size, which is Armstrong range. So I go with a more complex level, and hopefully you can understand generally what we're doing in our lab. We are interested in understanding how proteins signal from cell surfaces, and more specifically, how these signalings are mediated by lipids. How do you move this forward? I guess this way. Okay. So these are some of the current projects in the lab that involve all these cellular events. And in all cases, the signaling pathways are mediated by lipid-binding proteins. And more specifically, models regions in proteins that associate to lipids, and these associations trigger signaling pathways. So here are some of the proteins. Of course, I have a few minutes. So in order to make it simple, what I'm going to do is describe one of the major projects ongoing in the lab, and that represent pretty much what this picture is about. So I want to talk about proteins that are associated to endosomal protein trafficking. And this is something important for the cell, because cells, what you need to do is, when they don't need proteins at the cell surface, they need to get rid of them. Otherwise, you have uncontrolled cell signaling pathways. In order to do that, cells have different ways to remove proteins from the cell surface. So what my lab is studying is how these proteins are, what are the pathways by which these proteins follow, and how actually these proteins can be degraded in other compartments. So more specifically, we study what's going on in these compartments, which is called endosome. And on the surface of these compartments, there are proteins that what they do is collect the information or collect the proteins that are taken from the cell surface and continue the traffic for protein degradation. And this is the way that the cells are able to get rid of these proteins in case the cells don't require this type of signaling. So, and if you zoom in here, we study specific proteins that are modular in nature and require not only certain proteins that bring those proteins at the cell surface to these compartments, but also requires lipid signaling. So lipids, what they do in cells at the end is they provide directionality. They provide where these proteins should go in order to be degraded. Therefore, we study, for example, modular proteins, proteins that they are contains regions that specifically involved in a function. For example, regions are involved in association to endosomes, regions that are involved to bringing these proteins from the cell surface for degradation, or associating with other proteins, I can skip all this, that are typically located in other compartments, but when they are recruited, they recruit other proteins. They all together to exert that particular function. So I will skip all this because we don't have much time. So what I see here is all these proteins, what they do is recruit other proteins. And among other proteins are all these proteins are modified by certain signals. And these proteins are modified by certain signals, actually the proteins that need to be degraded. So what these proteins are doing is bringing these proteins and deliver them for degradation, for elimination at the intracellular level. So what we do? What we do is we study protein-protein and protein-lipid interactions at very high resolution, at atomic resolution. In order to do that, we study interactions using mainly solution nuclear magnetic resonance spectroscopy. We have an excellent instrument on campus that pretty much we are the major users. And in addition to that technology, we also use other biophysical approaches. So what is useful from these studies? You can actually look at how these interactions take place at very high resolution. Why you need that resolution? Because you can actually predict how the interactions take place and manipulate these interactions for undesired situations. For example, for drug design. So we use, for example, protein-lipid binding assays to monitor how these interactions take place. We look at interactions in real time, and also we using immobilization proteins in sensor chips, and we look at resolution of, or we look at interactions at very high resolution, very high affinity or low affinity in our lab. And also we can predict, for example, whether proteins can associate to different domains or not, and different regions. And so we can do that by using NMR spectroscopy. We map binding sites. We look at individual residues. That's the beauty of this technology. We don't actually do random or guessing how the interactions take place. We look at atomic resolution. That's the major tool in our lab. And here you can actually see regions at which proteins interact with other proteins, and you can map the binding site and see whether they overlap or not, like in this case. So also we can, because we use NMR spectroscopy, I have a few more minutes left. Okay. We also get the solution structure of proteins, and how useful is this information? Well, you can actually predict where the binding site is. In this case, it's a protein that is completely unfolded, is completely disordered, and somehow adopts a certain folding when associated to another protein. And therefore, with NMR spectroscopy, you can actually look at where the binding site is and predict how these interactions take place. And therefore, based on that information, you can actually map the site at which these interactions takes place, like in this example here. Also, with these associations, because we look at atomic resolution, you can replace the units of these proteins by others. These units are amino acids. And therefore, in this case, for example, we predict that a protein, when associated to another protein, inhibits lipid binding. And this is important because this association is critical for association of these proteins in the somal compartments. And using the NMR tools, actually you can replace these amino acids by others and abolish that. So with this technology, we can do that. And then based on these interactions, we can actually design models. Like I have a short movie here This is made by Rachel Robinson, so I appreciate her help on this, in which we can predict how proteins interact with membranes, how these proteins to be degraded contact these protein these compartments, and then how these proteins are incorporated in these compartments, how other proteins come and rearrange this organization based on our NMR studies and our atomic resolution studies. And with these associations, we can predict how signaling occurs in endosomal compartments. So I will appreciate, I also would like to appreciate the contribution of everyone in my lab. I'm not working with girls only, this is just by random, okay? But this is the girl group, and I'm bringing actually 2 males this semester, so to compensate the ratios. But I appreciate all the work from these students in my lab, collaborators all over the country, and funding for support. Thank you.
I'll start.
Go, Alan.
When you brought up the idea or paradigm of an engineering perspective on how cells are designed to accomplish the balanced goals of reliability in the face of complexity. Another aspect of cell magic is redundancy, and how it's amazing that cells can get along with what they need to do with this gene or that gene knocked out, and there's a small list of genes that in fact will be completely lethal, but a lot of genes can be, sort of optional. And I don't know, does that come into your modeling or perspective on analyzing biology?
Yeah, certainly. That's one of the aspects, or that's thought to be one of the huge factors for reliability, having this redundancy, actually. Yeah, so that's certainly an important factor. What also comes into play, I think, when you think about engineering versus how cells are made is the evolutionary history. I think an interesting question is how much of it is just a byproduct of like how cells evolved and how much of it is really needed. And I think synthetic biology will actually be nice to address that, because you can try to kind of make it simpler. You can try to apply the human engineering approach to cellular function and see how well it works. And that will partly answer whether that whatever we see in terms of complexity is really needed or whether it's a byproduct.
So I'm viewing this meeting really as a way for us to all collaborate and learn more about each other's work. So a question that I wanted to ask all of you, and I think Silka said this, is viewing these systems as integrated networks. Can you elaborate a little bit more of what sort of network techniques you've used to do structure analysis or the dynamics on these networks?
I can get started. So as I said, the question that we have is how proteins signal from cell surfaces. And we use a wide range of biophysics technologies that involve not only studies at the structural level, but also the functional level. And we go beyond that point. Also, because sometimes when you do in vitro experiments, you need actually to demonstrate that these findings can be reproduced in a physiological condition. So we use other technologies. I haven't shown those technologies in my presentation, but we also use studies in cell-based on organismal levels. And also, we collaborate with other groups on campus and outside of campus regarding how these interactions take place. And we do also bioinformatic analysis by molecular dynamic simulations to predict not only how the interactions take place, but also how to do modifications of those interactions and predict how these interactions may occur. And then based on these findings, that accelerates our research work, and then we can do those changes at the in vitro level.
Yeah. Well, so in terms of our research, there are 2 areas that, because I was fooling around, I didn't really have the time to explore. But I think, the connection between all these molecules. I mean, let me put it this way. The circadian molecules recognize and control the expression of genes based on recognition of specific elements, the regulatory region of genes that are very promiscuous in the genome. So they basically control the expression of very many genes. They also regulate interaction with other proteins. Now, the problem that we have in my lab is that we can check one thing at a time. So that is very minusculous, that is very reductionist, and that is a tremendous failure that molecular biology had in the last 20 years by trying to understand big processes by looking at what happened with one single gene and one single mutation. So network analysis, genomic analysis is not something fancy, something that we need these days to fully understand what's going on in a cell and the cell in the context of other cells in much larger environment. So we welcome all this analysis that can shed light on what's going on in these cellular processes.
I think we talked about this a little already today. It's a lot about the friction between biology and models, actually, how we can really fit them best. We have constant concerns about, how large a model should we take? How many components do we want to have in there? Do we just want a toy model, or do we want something mechanistic? Then the mechanistic thing or statistical models come with a completely different set of problems. So we try to match it with the question that we have, but it's actually really, it's something that needs permanent working on. It's actually almost a research in itself, like how do you optimally design your...
Yeah, I think biologists have been very arrogant in the past by considering that large data analysis was a fishing expedition. I think we pay for that arrogancy by ignoring a lot of data that was published before that really will help us to build new hypotheses based on that data. So hopefully our generation will prove the previous one wrong, as usually happens. Power.
I guess it's one for Carla. There was a major paper in Science by Johns Hopkins group claiming that, I'm not trying to play down the significance of environmental factors in cancer, but the conclusion of the paper was that the majority of cancers are bad luck.
I read the paper.
So I'm just curious of your opinion on this.
Yeah, I remember reading the paper. Actually, I passed it to everybody in my group. Immediately it was published. Well, what can I say? It was a good statistical study. It has been criticized, of course, by the scientific community. Is based on the sequence of very many genomes, and there is always a component of bad luck on getting cancer. There is a genetic predisposition for some cancers. There are some driver mutations that are critical for cancer. There are 14 of them that if they are there, you have higher propensity, and I think that's what they meant. I don't think they disregard other factors. I mean, I think the scientific community took it in a very, bad way. I mean, it's like a defensive way. And maybe because the title was, a little bit, I mean, it was sexy, but, but maybe a little bit challenging. But there is a component.
Okay, with that, if we can thank the VBI Biology Fellows for some great presentations.